Okay, let's dive in. Right now, as you're listening, your body is doing something pretty staggering. It's churning out, what, literally hundreds of billions of new blood cells.
That's right, every single day. It's this immense, ongoing construction project happening inside us all the time.
And the key players, the real orchestrators behind it all, are these fascinating cells called hematopoietic stem cells, HSCs.
Absolutely. They're truly remarkable. They're the ultimate source, the origin point for all the cells in your blood. Your red cells, white cells, platelets, the whole system.
And the text we're digging into today points out their influence goes even further, right? Affecting some cells and tissues outside the blood.
Yeah, exactly. Their main job is the blood, but they contribute elsewhere too. They are just fundamental for keeping us alive and functioning.
It really makes you wonder, doesn't it? How do they actually do that? How can such a, well, relatively tiny population of cells generate this unbelievable number of diverse, specialized cells every day?
And not just that, but how do they keep it up for an entire lifetime? Decades. What's the secret to their persistence?
Right. And crucially, why are they so central in medicine? We hear about them in transplants, saving lives, but also implicated in really serious diseases like leukemia.
Well, that's what this deep dive is all about. We're going to really get into the weeds of HSC biology, drawing heavily from this detailed source text that focuses specifically on them. We'll unpack their identity, their unique functions, the environment they operate in.
And the goal for you listening is to really get a solid handle on this foundational knowledge. We want to explore the core concepts, the how and the why, so you feel as familiar with HSCs as, the professionals working in this field. Let's start at the very beginning then. What fundamentally defines an HSC?
Okay. At the most basic level, hematopoietic stem cells, HSCs,
Yeah.
are the origin. They're the stem cells from which every single type of cell floating around in your peripheral blood arises.
Every last red cell carrying oxygen, every immune cell fighting off bugs, the platelets for clotting, all of it.
All of it traces back to an HSC. And as we mentioned, the source material even notes their contribution to specific cell populations in other tissues too. Their reach is significant.
That really highlights how indispensable they are. I mean, the source describes a pretty grim scenario if they're lost.
It does. If functional HSCs are wiped out, maybe by intense radiation or something similar, survival is measured in weeks, maybe 2 to 4, unless you have some pretty extraordinary medical intervention.
Wow. That starkly underlines their absolute necessity. You just can't live without them functioning properly.
It's a very clear demonstration, yeah. And on the flip side, their power is shown in how we use them clinically. Stem cell transplantation, basically giving someone a new, healthy HSC system, saves, what, tens of thousands of lives annually?
Yeah, the text mentions that figure. Treating blood cancers, genetic disorders. It's incredible.
It really is. It highlights their regenerative capacity.
But it's still kind of mind-boggling when you consider how few of them there apparently are. The source estimates only about 100,000 HSCs in an adult.
That's the estimate, yeah. Around 100,000 cells. And that small pool is responsible for generating hundreds of billions of mature cells every single day.
The sheer scale of that production from such a limited starting population is, well, it's astronomical.
It absolutely is. And that disparity drives so much of the research. How is that production rate even possible? How do they avoid just, burning out?
And the longevity question, how do they persist and function for decades, our entire lives, essentially?
These are the big fundamental biological questions that researchers are constantly chipping away at, trying to fully understand the mechanisms.
And the source suggests that studying this process, hematopoiesis, the making of blood, is actually a really important model for understanding other parts of the body too. Why is that?
Well, the blood system has some unique advantages for study. For one, it's pretty accessible. You can get samples relatively easily through blood draws or bone marrow biopsies if needed.
Easier than, say, brain stem cells.
Much easier, yes. And the cellular hierarchy, the path from the stem cell down through various stages to the final mature cell, is probably one of the best understood and most clearly defined developmental pathways in the body.
So it provides a clear roadmap.
Exactly. It serves as an excellent model system to figure out the general principles. How do stem cells maintain themselves? How do they generate specialized progeny? How is it all regulated? The lessons learned in hematopoiesis often provide insights into how stem cells work in skin, the gut, muscles, other tissues.
So it's like the foundational case study for stem cell biology.
You could definitely see it that way. And the research methods developed here are now pretty standard across the field. The text mentions things like defining the step-by-step molecular changes, identifying specific markers or flags on the cell surface that occur as cells mature.
Using those markers to sort and study specific population.
Right. Aggregating cells based on these markers, focusing research efforts on these really rare populations like the HSCs themselves. And a huge amount of what we know comes from functional tests, especially in animal models.
Like transplanting cells and seeing if they can rebuild the whole system.
Precisely. These reconstitution studies are critical. Can a single cell or a small population give rise to the entire blood system in a recipient animal? That's the gold standard functional test for a true HSC.
So we're assembling the picture from these different lines of evidence markers, function, observation.
Exactly. But it's important to keep in mind, as the source text points out, that our knowledge, especially about those deepest, most dormant stem cells, is still often based on inference.
We see what they produce, but not always the cell itself in action.
Correct. We can observe their descendants, we can do experiments that imply certain behaviors, but directly watching a single human HSC divide in its natural niche over, say, a year. That's still technically extremely challenging, if not impossible currently.
Right, given how rare and hidden away they are. Okay, so let's get to their core jobs. The source mentions two cardinal functions. What are these essential abilities?
These are the defining characteristics shared by all true stem cells, not just in the blood system. They are self-renewal and differentiation. The text we're using even references figure 96-1, which shows this basic concept.
Self-renewal, that sounds like the key to their persistence, their longevity.
It absolutely is. Self-renewal is the ability of a stem cell to divide in such a way that it produces at least one daughter cell that is essentially identical to the parent stem cell, maintaining the stem cell pool itself.
Without that, they'd just run out.
Exactly. If every time a stem cell divided, both daughters went off to become something else, the stem cell pool would vanish very quickly. Tissue maintenance would fail. So self-renewal is absolutely essential to sustain the source over time.
And the other function, differentiation, that's about making all the different types of blood cells we actually need.
Precisely. Differentiation is the process where the stem cell, or its immediate offspring, commits to becoming a more specialized cell type and progressively matures into a functional unit for the tissue.
So making red blood cells, neutrophils, lymphocytes, platelets.
Yes, all the mature working cells that perform the tasks required by the blood and immune system. This process is vital. If differentiation is blocked or goes wrong, if cells can't mature properly, maybe they get stuck halfway, tissue function suffers badly. This can lead to things like bone marrow failure or even contribute to the development of cancers like leukemia.
And the fact that mature blood cells have such different lifespans must really emphasize the need for this continuous production line.
Absolutely, it's a key point. Some mature blood cells are incredibly short-lived. Neutrophils might only last hours or a few days, especially during an infection. Red blood cells circulate for around 120 days, 4 months or so.
Even memory cells, which last longer.
Right, memory T and B lymphocytes can persist for years, even decades, providing long-term immunity. But the sheer volume of daily turnover, especially for cells like neutrophils and red cells, is immense. The HSCs are that constant, reliable source, replenishing all these lineages continuously.
So when an individual stem cell divides, how does it decide? Does it make more of itself, or does it make cells that will differentiate? How does it balance those?
The text lays out 3 main possibilities for the outcome of a single HSC division. 1. Symmetric self-renewal. The stem cell divides, and both daughter cells remain stem cells.
Yep.
This expands the pool.
Okay.
2. Symmetric differentiation. The stem cell divides, and both daughter cells are committed to differentiate, leaving the stem cell pool unchanged in size for that division event, but increasing downstream production.
Right.
And 3. Asymmetric division. This is often seen as the classic model. The stem cell divides into 2 different daughters. One stays a stem cell, maintaining the pool, and the other embarks on the differentiation pathway.
That asymmetric division seems like the neatest way to keep things stable. One replaces the parent, one goes off to work.
It does feel intuitive for steady-state maintenance. But what's really interesting, and the text emphasizes this, is that the balance between these 3 modes isn't necessarily fixed. It's dynamic.
It can shift based on need.
Exactly. During development, when the whole system is being built, symmetric self-renewal might be favored to rapidly expand the initial stem cell population. Conversely, if there's a sudden high demand, say, after significant blood loss or a major infection, the system might shift towards more symmetric differentiation to quickly boost output. This flexibility is key to the system's responsiveness.
You mentioned development. That whole journey of where blood is made sounds incredibly complex. Where does it even start in an embryo?
It's a fascinating migratory story outlined in the source. The very first blood cell production doesn't even happen inside the main body of the embryo. It starts in the yolk sac, an extra embryonic structure.
Outside the embryo itself, what does it make there?
Primarily, it makes embryonic red blood cells, which are structurally a bit different from adult red cells, and also the founding population for certain types of macrophages that take up residence in tissues long term, like the microglial in the brain.
Wow. So the brain's immune cells originate way back then from the yolk sac.
That's the current understanding, yes. After the yolk sac phase, the center of action shifts. It moves sequentially to the placenta and also some temporary sites within the developing embryo.
And then?
Then, for a significant chunk of fetal development, the liver takes over as the major site of hematopoiesis. The fetal liver is buzzing with blood cell production.
And finally, it settles in the bone marrow.
Right. Later in gestation, typically in the second trimester, the definitive long-term sites get established. That's primarily the bone marrow, inside the newly forming bones, and also the spleen plays a role.
Does the type of blood cell produce change depending on where it's being made?
Yes, absolutely. The output is tailored to the site and developmental stage. Yolk sac, embryonic red cells, specific macrophages, intraembryonic sites. This is where the first true HSCs capable of long-term repopulation emerge, along with precursors for adult-type red cells, platelets, and innate immune cells.
And the bone marrow and thymus.
Once they're colonized and functional, they become the main production centers for the adaptive immune system. The T lymphocytes maturing in the thymus, and B lymphocytes developing primarily in the bone marrow. These give us that specific, long-lasting immunity.
And I imagine the stem cells must be dividing like crazy during all this development.
Extremely rapidly, yes. Stem cell proliferation rates are very high throughout fetal life and remain elevated for a little while after birth as the system scales up. But then, the text notes, there's a pretty dramatic slowdown. The proliferation rate drops significantly as the system transitions to the adult, more stable, and largely quiescent state.
How do the stem cells actually get into the bone marrow from, say, the fetal liver? It's deep inside bone.
They travel through the bloodstream. Cells produced in the fetal liver, or these other sites, enter the circulation. Once the long bones start to mineralize and form cavities, typically starting around the second trimester, these circulating HSCs can then migrate out of the blood vessels and into these newly forming marrow spaces.
Homing in on the right spot. But you mentioned earlier that HSCs circulate even in adults. Is that just leftover movement, or is it ongoing?
The source confirms it's ongoing throughout life, not just a developmental relic. However, the time an individual HSC spends in the blood seems very short, potentially just minutes, based on mouse studies.
But even that brief transit is important.
Functionally, yes. Because even these circulating HSCs, if collected, are fully capable of homing back to the bone marrow niche and reestablishing long-term hematopoiesis. That's the basis for transplantation using peripheral blood stem cells.
Right, which leads directly to how we harvest them clinically. We exploit the circulation.
Exactly. We don't always have to do painful bone marrow harvests anymore. We can use a process called mobilization to coax more HSCs out of the marrow and into the bloodstream temporarily.
Increasing their numbers in circulation?
Significantly, yes.
Yeah.
Then clinicians can collect these mobilized peripheral blood stem cells using a machine in a process called leukapheresis. It's much less invasive for the donor or patient.
So how does that mobilization work? How do cells physically get out of and back into the marrow? It must involve specific molecular interactions.
It's a very precise multi-step process, like a molecular docking procedure. The text describes it well. When a circulating HSC enters the marrow's blood vessels, it first needs to slow down.
How does it do that?
It uses adhesion molecules on its surface, like CD162 and CD44, to make transient rolling contact with molecules called selectins, P-selectin and E-selectin, on the surface of the endothelial cells lining the vessel. This interaction acts like gentle brakes, causing the cell to roll along the vessel wall.
Like Velcro, almost. Light catch and release.
That's a good analogy, yes. Then for a firm grip, other adhesion molecules called integrins on the stem cell get activated. A really key interaction is VCM1 on the stem cell binding strongly to VLA4 on the vessel wall. That stops the rolling and makes the cell adhere firmly.
Okay, so it's stuck to the wall. How does it get through the wall into the niche?
Chemical signals guide it. A crucial chemokine, a small signaling protein called CXCL12, also known as SDF1, is highly concentrated in the bone marrow niche. Stem cells have the receptor for it called CXCR4. This CXCL12-CXCR4 interaction acts like a powerful attractant, drawing the cell towards the source. There are other signals, too, involving ionic calcium sensing. These signals guide the cell to migrate across the vessel wall and into the marrow tissue.
And this CXCL12, CXCR4 interaction is important not just for getting there, but for staying there.
Absolutely critical. The source highlights it's essential for the initial homing during development, like from the fetal liver to the marrow. And, very importantly, it's a major factor in retaining HSCs within the bone marrow niche in adults. It tethers them there.
Ah, so that's the target for mobilization. If you want to get HSCs out into the blood for collection, you need to disrupt that CXCR4, CXCL12 tether.
Precisely. That's the core strategy behind clinical mobilization protocols. We use drugs that interfere with this interaction.
How?
You can block the CXCR4 receptor on the stem cell directly, preventing it from seeing the CXCL12 signal. Or you can use agents that cause the CXCL12 in the niche to be degraded or altered, weakening the signal. Or sometimes the treatments cause the stem cell to internalize its own CXCR4 receptors, making it less responsive. Any of these approaches effectively loosens the stem cell's anchor to the niche.
And releases it into the bloodstream.
Exactly, allowing it to be collected via leukapheresis.
What drugs are actually used for this?
The known ones mentioned in the text are GCSF, granulocyte colony stimulating factor, which works indirectly on this axis, and plerixafor, which is a direct blocker of the CXCR4 receptor. They are standard tools for HSC mobilization.
It's amazing how that detailed molecular understanding translates directly into a procedure that helps thousands of patients.
It really is a fantastic example of bedside translation. And the text suggests that digging even deeper into these mobility mechanisms, how cells enter, leave, find specific spots within the marrow, could lead to even more efficient mobilization techniques, or perhaps ways to improve how transplanted cells find their new home.
Their home, the niche. Let's unpack that concept more. It's not just a physical space, right?
No, absolutely not. The idea of a specialized microenvironment, the niche, is really central to stem cell biology. It was first proposed way back to explain why transplanted bone marrow cells seem to know where to go and what to do. The niche is an anatomical location, yes, but its crucial function is providing regulatory signals to the stem cells living there.
What kind of signals? What does the niche actually provide for the HSC?
It plays several vital roles, as the text describes. First, it provides the essential support factors that allow the stem cells to survive, maintain their stemness, and basically thrive long-term.
Keeping them healthy.
Right. Second, it permits them to expand, to undergo self-renewal division, when the body needs to increase the stem cell pool, maybe during development or recovery. Third, it instructs them on how many and what type of differentiating descendant cells to produce to meet the body's current needs.
So it tells them when to divide and what to make.
In a sense, yes. But crucially, the fourth function is regulation, or constraint. The niche also provides signals that limit stem cell proliferation and production. It prevents them from growing uncontrollably.
Ah, so it's nurturing but also keeps them in check, prevents them from going rogue.
Exactly. The text describes it perfectly as a home that is both nurturing and constraining. It gives them what they need, but also imposes rules to maintain balance and prevent dangerous overgrowth, like cancer.
Does the niche location also shift during development following the HSCs?
Yes, it mirrors that journey. There's a niche environment in the yolk sac, then the fetal liver, the spleen, and finally the bone marrow becomes the primary adult niche.
And within the adult bone marrow, where exactly is it? Is it everywhere or specific spots?
The source suggests it's strongly associated with the blood vessels, the vasculature, particularly in the trabecular or spongy bone regions. There's also evidence pointing to the endosteal region, the surface right next to the bone itself, as being particularly important.
Why the endosteal region specifically?
It might be especially crucial for transplanted cells, potentially because the conditioning treatments given before transplants can damage the more central marrow areas, leaving the endosteal surface as a relatively protected haven where cells can engraft.
Makes sense. So what's actually in the niche? What cells and molecules create this special environment?
It's a complex community. The text highlights mesenchymal stromal cells. These are connective tissue support cells and the endothelial cells that form the blood vessel walls as key players. They produce essential factors like KIT ligand and that chemokine CXCL12 we keep mentioning, which are vital for HSC maintenance and retention.
Are other cell types involved too? It sounds like a busy place.
Very busy. The list in the source is quite extensive. Other cells implicated in regulating HSCs include components of the nervous system found in the marrow, like sympathetic neurons and their associated Schwann cells. Immune cells, like macrophages, play a role. Even cells involved in bone turnover, megakaryocytes, platelet precursors, osteoclasts, bone resorbers, and osteoblasts, bone builders, seem to influence the stem cells, either directly or indirectly.
Wow, it's a whole ecosystem.
It truly is.
Mm.
And it's not just cells. The extracellular matrix, the non-cellular scaffold made of proteins like osteopontin and sugars like heparan sulfates, also provides important structural and signaling cues to the HSCs.
So understanding this complex niche environment is critical for medicine.
Absolutely essential. Maintaining a healthy niche is vital for normal blood production. And for transplantation, ensuring the transplanted HSCs can find and interact properly with a supportive niche is a major factor determining success or failure.
And can the niche itself contribute to disease?
Yes, and this is a really important point highlighted in the text. It's an active area of research. Experimental studies have shown that if you introduce mutations specifically into the niche cells, not the stem cells themselves, it can actually drive the development of myeloid cancers like leukemia in animal models.
So the seed might be okay, but the soil can become corrupted and cause problems.
That's a perfect analogy.
Yeah.
It tells us the niche isn't just a passive bystander. It can actively participate in the disease process. This opens up really exciting therapeutic possibilities. Maybe we can target the niche to help treat blood cancers or to better support healthy stem cells in other conditions.
Let's go back to that incredible production capacity. You said we basically never run out of HSCs normally. The source calls this excess capacity. How much excess are we talking?
It's really quite remarkable. The text stresses that, barring disease, our HSC pool is more than sufficient for our entire lifespan. The evidence for this is compelling.
Like those mouse transplant studies.
Exactly. Serial transplantation experiments in mice, where marrow from one animal can successfully reconstitute the blood system of several consecutive recipients, show there's far more functional potential than needed for just one lifetime.
And we see this in human transplants too.
We do. People who receive stem cell transplants from a donor often maintain healthy blood counts for decades, relying on that initial graft. This works even though the number of HSCs actually transferred in a transplant is likely only a fraction of the total pool in a healthy person. It demonstrates a substantial reserve capacity.
That's amazing redundancy. But they're still regulated. How do they know when to ramp up production, say, if you need more red cells after bleeding or more white cells for an infection? How do the stem cells get that message?
You know, it's interesting. The text points out that how the stem cells themselves directly sense and respond to these kinds of systemic demands is actually still not fully understood. We know a lot about how the downstream cells are regulated.
Like with EPO for red cells.
Right. There are negative feedback loops. Low red cells trigger EPO production by the kidney, which stimulates red cell precursors to divide and mature. Low neutrophils might trigger GCSF release, which drives granulocyte precursors. These are well-established pathways for the committed progenitors.
But you mentioned GCSF doesn't really make the stem cells divide faster directly.
That's the crucial distinction the source emphasizes. Many of these potent growth factors that strongly stimulate proliferation in the progenitor and precursor stages seem to have little or no direct effect on the cell cycle speed of the most primitive quiescent HSCs. GCSF, for example, its main effect on HSCs, seems to be altering their adhesion to the niche, causing mobilization, rather than directly pushing them into division within the niche.
So the signals controlling the stem cell pool must be fundamentally different from the signals controlling the factory floor, so to speak.
That appeared to be the case. It's a really key concept. The text gives examples of molecules and pathways that seem specifically important for HSC survival, self-renewal, or quiescence, but which have different, or sometimes minimal, effects on the more mature progenitor cells.
Like what kind of molecules?
Things like specific cell cycle inhibitors that keep HSCs dormant, certain transcription factors that control HSC-specific gene programs, enzymes involved in RNA processing like dicer, even differences in metabolic regulators like pyruvate kinase isoforms. These seem to operate at the stem cell level.
It really drives home the point that HSCs are a distinct entity with their own unique set of rules.
They absolutely are. And understanding these distinct regulatory mechanisms is vital because they offer potential therapeutic targets. You could potentially influence the stem cell pool specifically without causing unwanted massive proliferation downstream, or vice versa.
Okay, let's visualize that process again, how one HSC gives rise to everything. You mentioned the hierarchy, the differentiation tree. Can you walk us through that again, maybe thinking about figure 96.2 from the source?
Sure. Imagine a tree. At the very bottom, the root and trunk. That's the hematopoietic stem cell. multipotent, capable of becoming any blood cell, long-lived, potentially lasting years or decades, and crucially, capable of self-renewal. That's the foundation.
Then the first major branches splitting off.
Those represent the early progenitor populations. These cells have already lost some potential compared to the HSC. They're often described as being committed to broad lineages, like the common lymphoid progenitor, CLP, giving rise to B cells, T cells, NK cells, or the common myeloid progenitor, CMP, leading towards red cells, platelets, granulocytes, and monocytes.
And these progenitors are the workhorses of proliferation.
Largely, yes. They are generally shorter-lived than HSCs, but they divide much more rapidly. The text calls them transient amplifying cells. Their job is to take the relatively small output from the slow-dividing HSCs and massively expand the number of cells committed to a particular path.
So most of the cell divisions actually happen here, not at the stem cell level.
That's right. The major amplification happens in the progenitor compartment. Then, moving further up the branches, you get to the precursor cells.
More specialized still.
Yes. These are typically committed to just a single lineage. Think of an erythroblast, which will only become a red blood cell, or a myeloblast, destined to become a granulocyte. They are still dividing, but they're further down the maturation path and are starting to acquire specialized features, though they aren't fully functional yet.
And finally, the leaves on the tree.
Those are the fully mature, differentiated cells. These are the end products, the red blood cells, neutrophils, lymphocytes, platelets, etc., that are released into the blood to do their jobs. They're typically terminally differentiated, meaning they don't divide anymore.
How does a cell actually make these choices? Moving down the tree and picking a branch, what guides it?
It's a combination of factors. There are internal changes, shifts in the cell's gene expression patterns, controlled by key transcription factors, some of which are listed in Figure 96-2. And there are external cue signals from the niche environment, soluble growth factors, and cytokines, like those shown in blue in the figure. It's the interplay between this intrinsic program and the extrinsic signals that dictates the cell's fate decisions. Our understanding of the full network is still incomplete, though.
And as cells move down this pathway, what fundamentally changes about them?
Two main things. Functionally, they acquire the specific tools and machinery they need for their final job, making hemoglobin, developing granules, expressing antigen receptors, etc. But crucially, they also progressively lose plasticity.
Meaning they become less flexible in their choices.
Exactly. A common myeloid progenitor has several potential fates open to it. But once it commits to becoming, say, a megakaryocyte precursor, it normally can't just switch and decide to become a neutrophil instead. That pathway is largely locked in. The text does mention an emerging idea of a lymphomyeloid potent progenitor, which might sit very early, complicating the simple two-branch split, suggesting maybe the decisions are a bit more fluid right at the top.
But generally, it's a one-way street towards specialization.
Under normal circumstances, yes. Once a cell passes a certain commitment point, it doesn't typically reverse or switch lineages. Although, as the text notes, artificial reprogramming using genetic techniques can force cells to change identity, showing the potential is still encoded, just silenced.
And the ability to divide usually decreases as they mature. Figure 96-3 seems to show that.
That's the general trend, yes. As cells become more specialized and terminally differentiated, they typically lose their proliferative capacity. Mature granulocytes, for instance, are non-dividing. Their numbers are maintained purely by ongoing production from earlier precursors.
Are there exceptions to that rule?
A couple are mentioned. Certain tissue-resident macrophages can apparently undergo some local proliferation. And as we discussed, lymphoid cells, T and B cells, retain the ability to proliferate significantly. But that's usually triggered by a specific event, like encountering their target antigen during an immune response. It's not the same as the continuous baseline production from progenitors.
So the real engine room for generating the massive numbers of cells needed daily is that transient amplifying progenitor compartment?
Precisely. It's a common strategy in tissues with high turnover, like the gut lining or skin as well. You have a quiescent stem cell pool providing the source, and a highly proliferative intermediate compartment doing the bulk manufacturing. This protects the precious stem cells from excessive division.
And how long does that manufacturing process take, from a committed progenitor to a fully functional cell ready for duty?
The timing seems pretty fixed. The text suggests it takes roughly 10 to 14 days in humans for a progenitor cell to go through all the maturation steps and become a functional cell released into circulation. You can see this time frame reflected in how long it takes blood counts to recover after chemotherapy wipes out the progenitors.
So there's always a built-in lag of about 2 weeks in the production line. Interesting. You also touched on the idea that maybe not all HSCs are created equal in terms of what they tend to produce, this concept of biased stem cells.
Yes, this is challenging the older, simpler view. The classical model often depicted HSCs as a uniform population, all equally capable of producing all lineages. But newer evidence, highlighted in the source, suggests there's heterogeneity within the HSC pool itself.
Meaning individual HSCs might have preferences.
Kind of. Some individual HSCs might be intrinsically biased or predisposed to generate more cells of one particular lineage, say, producing more myeloid cells than lymphoid cells, or vice versa compared to other HSCs in the same pool. It adds another layer of complexity to how the balance of blood cell production is maintained.
Fascinating. Suggests the stem cell pool might be more structured or diverse than we thought. Let's really focus now on self-renewal. That ability to make more of themselves, how is that capacity managed?
Okay, so an HSC faces these critical fate choices: die, self-renew, or differentiate. That ability to divide and reliably produce another functional stem cell self-renewal is really special. Only stem cells, and those memory lymphocytes we talked about, seem to robustly combine proliferation with maintaining their own population identity long-term.
Was it always thought that self-renewal was exclusively an HSC property within the blood system, apart from lymphocytes?
That was the dominant view for a long time, yes. Once a cell started differentiating, committed to a lineage like myeloid, it was thought to have lost self-renewal capacity. However, the text mentions that more recent studies suggest some committed myeloid progenitors might actually retain a limited ability to self-renew, at least for a while, in vivo.
So the boundary might be a little less absolute than we thought.
Potentially, yes. It suggests some downstream progenitors might contribute more sustainingly to their lineage than previously appreciated. It's an evolving picture.
Are all the adult HSCs equally active, or are some just sitting back?
The text is very clear. They are not all equally active. Adult HSCs, like stem cells and other tissues, are a heterogeneous bunch. There seems to be a spectrum. Some HSCs are thought to be in a state of deep quiescence or dormancy.
Meaning they barely divide.
Exactly. They might divide only once every few months, or perhaps even less frequently, maybe only a few times over many years. These are often considered the deep reserve pool, the ultimate long-term backup.
And others are more active.
Yes, there are likely other HSCs that cycle more frequently, maybe dividing every few weeks or months. These are probably the ones responsible for the more routine, day-to-day replenishment of the progenitor pool that drives constant blood production.
So like a reserve team and the players currently on the field.
That's a decent analogy. And these deeply quiescent HSCs are notably resistant to many external signals, like the growth factors, cytokines, that stimulate their more active descendants to divide. This dormancy protects them.
Is that dormancy why it's been so hard to grow HSCs in the lab? They just don't want to divide.
That's a huge part of the challenge, yes. Replicating that specific quiescent state, providing the right signals, or lack thereof, to maintain true long-term stemness while getting them to expand in number ex vivo, it's proven incredibly difficult. They often tend to either differentiate, die, or lose their long-term repopulating ability when cultured.
What keeps them locked in that quiescent state? What are the molecular breaks?
Several mechanisms are mentioned. Intrinsically high levels of proteins called cyclin-dependent kinase inhibitors, CKIs, seem key. Things like P57, also called CDKN1C. These proteins physically block the molecular machinery that drives the cell cycle, particularly the transition from the G1 resting phase into the S phase where DNA is copied.
So they put a stop sign on division.
Essentially, yes. And extrinsically, signals coming from the niche environment also play a role in enforcing quiescence. The text mentions factors like angiogenin, IL-18, and angiopoietin-1 as potential niche-derived signals that help keep HSCs dormant.
Does this regulation change as we get older? Does aging affect HSC quiescence and self-renewal?
It appears so, yes. The text indicates that HSC function does change significantly with age. Both factors within the aging stem cells themselves, like an increase in another CKI called P16INK4A, and changes in the surrounding bone marrow niche, such as a decline in nerve signals from the sympathetic nervous system, seem to contribute.
So aging affects both the cell and its environment.
Right, and these changes are linked to alterations in HSC cycling, potentially skewing their differentiation output, often towards myeloid lineages, and sometimes impairing their overall regenerative capacity in older individuals.
That sounds like a natural decline. Can any of it be pushed back? Can we reverse those age effects?
Well, intriguingly, the text mentions experimental studies, mostly in aged mice, where manipulating some of these pathways showed promise. For example, reducing the levels of that age-associated inhibitor P16INK4A, or stimulating certain nerve signal receptors, beta-3 adrenergic receptors, in the marrow of older animals, actually improved HSC cycling and their ability to reconstitute the blood system upon transplantation.
Making old stem cells behave more like young ones.
Functionally, yes, in those specific assays. And importantly, these interventions seem to specifically affect the stem cells without drastically changing the overall numbers of mature blood cells, again pointing to distinct regulatory circuits.
That's really hopeful. It suggests that targeting these specific molecular pathways governing stem cell aging or function could be a therapeutic strategy down the line.
It certainly supports that idea. And understanding self-renewal is probably the most critical aspect for clinical applications, especially transplantation. The sheer number of functional, self-renewing HSCs you can collect and transplant is often the limiting factor.
For both using your own cells, autologous, and donor cells, allogeneic.
Yes. If we had reliable ways to control self-renewal, specifically to expand the number of true functional HSCs outside the body, that would be revolutionary for transplantation medicine.
How so? What would be the benefits?
Well, first, it could dramatically reduce the amount of cells needed initially. This would make harvesting less burdensome, less invasive, maybe less expensive for donors or patients.
And it could open up other cell sources that are currently limited by cell dose.
Exactly. The prime example is umbilical cord blood. Cord blood units are packed with high-quality HSCs, and they have advantages like lower risk of graft versus host disease and better availability for diverse populations. But often, a single unit just doesn't contain enough HSCs to successfully engraft an adult patient.
So expansion could make cord blood transplants much more feasible for adults.
That's the major hope. If you could reliably expand the HSCs from a cord blood unit tenfold, say, it could become a much more widely used option. Although the text does note that advancements in haploidentical transplants, using half-matched family donors, have provided another powerful option, somewhat reducing the pressure on cord blood expansion recently.
And what about gene therapy? Expansion seems crucial there, too.
Absolutely critical. When you're doing gene therapy, you often start by collecting the patient's own HSCs, correcting the genetic defect in the lab, and then transplanting them back. You need enough of those corrected cells to take hold and repopulate the entire system. Being able to expand the gene-corrected HSC population ex vivo before infusion would massively increase the chances of success in long-term cure.
So mastering self-renewal is key. But you said the exact molecular control is still murky. What clues do we have?
It's definitely an area of intense research. One major theme involves how DNA is packaged and regulated what's called the chromatin state. The way DNA is folded and chemically tagged affects which genes can be turned on or off. Certain protein families that control this chromatin structure seem crucial for HSC self-renewal.
Like which ones?
The text highlights the polycomb group proteins. These are complexes that modify chromatin to silence gene expression. Specific members like BE1 are known to be essential for HSC self-renewal, possibly by keeping genes that promote differentiation or cell cycle entry switched off.
So BMI1 helps maintain the stemness.
It seems to be a key part of its role, yes. Loss of BI1, or another important regulator called GFI1, leads to a reduction in HSC numbers and function. Another polycomb-associated protein mentioned is ASLS1.
And do these regulators go wrong in cancer, linking back to that idea?
Yes, very much so. Dysregulation is common in blood cancers. For instance, overexpression of BI1 can promote the self-renewal of leukemic stem cells.
Yeah.
And mutations in ASXL1 are incredibly frequent in diseases like myelodysplastic syndromes, MDS, and acute myeloid leukemia, AML, often contributing to the abnormal growth.
Are there other types of molecules involved in regulating self-renewal?
The text also points to the homeobox, or HOX genes. These are master regulators, famous for patterning the body during development, but some also play ongoing roles in adult stem cells. Experimentally, overexpressing certain HOX genes, like HOXB4, can induce massive self-renewal of HSCs. Other HOX genes are also implicated in normal HSC function and are often dysregulated in leukemia.
It really seems like the machinery for normal self-renewal is tightly intertwined with the pathways that get hijacked in cancer.
That's a very strong connection emerging from the research. The source also emphasizes epigenetic modifiers enzymes that add or remove chemical tags on DNA or its associated proteins, changing gene activity without altering the DNA sequence itself.
Like DNA methylation.
Exactly. DNA methyltransferase DNMT3A is mentioned, as is TET2, an enzyme involved in demethylation. Mutations in DNMT3A and TET2 are, like ASXL1 mutations, very common early events in the development of MDS and AML.
Early events. You mentioned clonal outgrowth before. Can you explain that again?
Sure, clonal hematopoiesis is where a single HSC acquires a mutation, say in TT2 or DNMT3A, that gives it a slight survival or growth advantage over its normal neighbors. This single mutated cell then starts to divide more or survive better, and its descendants, the clone, gradually take over a larger fraction of the total stem cell pool.
So one mutated cell starts to dominate.
Right. These mutations themselves often aren't enough to cause full-blown leukemia right away, but they create this expanded pool of pre-malignant stem cells. This clonal dominance significantly increases the risk that one of these cells will acquire the additional mutations needed to transform into leukemia. They're often called founder mutations because they establish the foundation for the cancer to develop later.
That link between normal stem cells and cancer seems undeniable. The source mentions the idea that cancers might be organized hierarchically, like normal tissues, with cancer stem cells at the top.
That's the cancer stem cell, CSC hypothesis, yes. It borrows from our understanding of normal tissue stem cells.
Yeah.
The idea is that even within a tumor, which might look like a uniform mass of bad cells, there could be significant heterogeneity.
Different types of cancer cells within the same tumor.
Potentially, yes. And the CSC model proposes that a small subpopulation within the tumor acts like the stem cells. They have the capacity for self-renewal to perpetuate themselves, and also the ability to differentiate and generate the more numerous non-stem cancer cells that make up the bulk of the tumor.
And these CSCs might be the ones driving tumor growth and relapse.
That's the implication. If CSCs exist, they might be more resistant to conventional therapies, which often target rapidly dividing cells. They might be slower cycling, and they might be the cells responsible for metastasis and treatment failure. Therefore, therapies would need to specifically eliminate these CSCs to achieve a lasting cure. Myeloid leukemias are one area where this concept has been studied quite extensively.
Does this mean that cancer always originates from a normal tissue stem cell?
Not necessarily, and the text makes this distinction clear. While the presence of cells with stem-like properties, self-renewal, differentiation potential, within a tumor is central to the CSC idea, it doesn't automatically prove that the very first cancerous cell was a normal HSC.
So a different cell could acquire those properties.
It's possible. Cancer development is usually a multi-step process requiring several genetic hits. It's conceivable that a more committed progenitor cell, or even a precursor, could acquire mutations that not only drive uncontrolled growth, but also reactivate or grant it the property of self-renewal, allowing it to function like a stem cell and initiate the tumor.
Why might normal stem cells be particularly susceptible targets, though?
Because they already possess two key characteristics needed for cancer development. Longevity, they present for years, and extensive proliferative potential. They can generate billions of daughter cells. This gives them ample opportunity to accumulate the multiple mutations required for transformation.
Has this been tested? Can we tell if cancers usually start in stem cells versus progenitors?
Yes, the hematopoietic system has been a key model for these kinds of experiments. Researchers can isolate distinct populations, HSCs, various progenitors, precursors using surface markers. They can then introduce specific cancer-causing genes, oncogenes, into each population and see which cell type is most efficiently transformed into a leukemia that can be serially transplanted, indicating it contains the self-renewing cancer-initiating cell.
And what's the verdict? Is it always the stem cell?
The results are nuanced, according to the source. It really depends on the specific oncogene being tested. For some oncogenes, the normal HSC population was indeed the most efficient cell of origin. But for others, more committed progenitor populations were found to be perfectly capable of initiating and sustaining the leukemia, implying they acquired the necessary self-renewal capacity during transformation.
So the crucial takeaway is that malignant cells can acquire stem cell-like properties regardless of their exact starting point.
That seems to be the key functional insight. The acquisition or dysregulation of self-renewal pathways appears critical for the persistence and propagation of many cancers, whether that pathway was inherent to the cell of origin or gained later.
We've talked a lot about blood and cancer. Does the source mention anything else these versatile HSCs might be able to do, going beyond blood?
It touches briefly on some areas that are more experimental or controversial. There has been some research, the text notes, suggesting that HSCs, or perhaps other cells from the bone marrow when infused, might contribute to healing damaged tissues, like in the heart after a heart attack or the brain after a stroke, perhaps by promoting blood vessel repair.
Is that a proven therapy?
The text characterizes this as largely experimental, and the data as somewhat controversial regarding how much these cells truly integrate and function as non-hematopoietic cells versus providing supportive signals. It's not considered standard clinical practice for those conditions outside of trials.
But could we potentially make them do other things using reprogramming?
Now that's a different angle with huge potential. Because the HSCs are relatively accessible, they are an attractive starting population for cellular reprogramming technologies.
Like iPSCs,
Yeah.
induced pluripotent stem cells.
Exactly. You could potentially take HSCs, reprogram them back to a pluripotent state, and then guide their differentiation towards entirely different cell types needed for therapy.
What kinds of cells are people trying to make from them?
The text mentions active research areas like generating mature lymphoid cells, like T cells, engineered for cancer immunotherapy. Another major goal is developing ways to produce large quantities of red blood cells or platelets in the lab from reprogrammed stem cells, which could eventually reduce our reliance on human blood donors.
That would be game-changing for transfusion medicine. Okay, finally, the source highlights HSCs as prime targets for gene therapy.
They really are perhaps the ideal target for many genetic diseases affecting the blood and immune system. The tools we have now for altering genes, changing sequences, tweaking expression levels, fixing errors are advancing at an incredible pace.
And because HSCs last a lifetime and produce everything?
Exactly. If you can successfully correct the genetic defect in a patient's HSCs, those corrected cells can then self-renew and differentiate, potentially providing a lifelong supply of healthy blood and immune cells. It offers the prospect of a one-time curative treatment.
What specific gene therapy technologies are being used with HSCs?
The text mentions several approaches. Viral vectors, using engineered viruses like lentiviruses or retroviruses to deliver a correct copy of a faulty gene, have been used successfully for treating severe primary immunodeficiencies, for example.
Are there non-viral methods too?
Yes, antisense technology is mentioned. This uses small molecules that can bind to RNA and block the production of a specific protein. A key example being developed is blocking the protein BCL11A.
Why block BCL11A?
Because BCL11A normally acts to switch off the production of fetal hemoglobin shortly after birth. In diseases like sickle cell anemia or beta thalassemia, where the adult hemoglobin is defective, blocking BCL11A allows the cells to start making healthy fetal hemoglobin again, which can compensate for the faulty adult form.
Reactivating a backup system, essentially. And what about the really precise editing tools like CRISPR?
Absolutely. CRISPR-Cas technology and similar genome editing systems are being actively investigated and used in clinical trials with HSCs. The goal here is not just to add a gene, but to directly repair the specific mutation in the patient's own DNA that causes the disease.
Wow, the precision is just getting better and better.
It really is. The text concludes by emphasizing that these precision genetic manipulation technologies are rapidly expanding, and the hematopoietic system, thanks to the properties and accessibility of its stem cells, is right at the heart of this therapeutic revolution.
So pulling it all together, these hematopoietic stem cells are truly foundational. They are essential for life itself, constantly rebuilding our blood and immune systems with this incredible capacity.
And possessing enormous potential for healing, as we see in transplantation, and are beginning to unlock with gene therapy. They are the ultimate regenerative engine in the blood system.
There's that duality. That same power, particularly the capacity for self-renewal, if it gets dysregulated through mutation or other insults, can become the source of devastating diseases like leukemia.
It highlights the incredibly fine-tuned balance required. Understanding how they work, how they're controlled by internal programs and external niche signals, is absolutely paramount for developing better therapies, whether we're trying to boost their function, replace them, or stop them when they go wrong.
And hopefully, through this deep dive, you listening have gained a much richer, more fundamental appreciation for these vital cells, going beyond just the headlines to understand the core biology, much like the experts who work with them every day. It really makes you think. Given their amazing resilience, their excess capacity, and the intricate layers of control we're still uncovering, how much more is there still to learn? What hidden potential or fundamental biological secrets are locked away in their dormant states or their complex niche interactions?
It's a great question. What new therapeutic avenues might open up, or what deeper insights into aging or cancer might emerge as we peel back that next layer of complexity? It's definitely going to remain a fascinating and vital area of research for years to come.